Hello everyone, welcome back. So this is the last week of our course, Fundamentals of Protein Chemistry. This course is coordinated by Professor Dasgupta from the Department of Chemistry, IIT Kharagpur, and I'm Beas Mukherjee, a research scholar from the Department of Bioscience and Biotechnology, IIT Kharagpur. And in today's class, we are going to talk about a lot of different topics since this is the last class for this session. Okay, so we are going to talk about different topics such as ROS, which is reactive oxygen species, and we are going to talk about intrinsically disordered proteins, viruses, and a little bit about protein sequencing. Okay, so let's begin with today's class. Okay, so we'll start with what exactly is ROS or reactive oxygen species. Okay, so reactive oxygen species are chemically reactive oxygen containing molecules that are naturally produced as a byproduct of cellular metabolism at high concentrations. ROS induces oxidative stress inside the cell, resulting in subsequent damage of the cellular macromolecules such as nucleic acids, lipid membranes, and cellular proteins. Okay, this is a normal part of the cellular process. Okay, this is a normal thing that happens inside the cell, but when this happens in an uncontrolled way, then what happens is the cell is killed. Okay, so around 30 to 40% of the modern day therapeutics and cosmetic products incorporate antioxidants or ROS modulating components, as oxidative stress underlines many diseases and aging processes. Okay, so one of the major ways that we age, that our cells age, is through the production of these reactive oxygen species. Okay, and a huge huge amount of the market, consumer products and therapeutics, all of these actually target reducing these oxidative damage in our cells so that aging can be slowed down, so that different diseases can be combated. Okay, now what are these different reactive oxygen species? So some of the examples are given over here. So hydrogen peroxide, superoxide radical, molecular oxygen is converted into singlet oxygen form. So all of these different forms are there, and how are they getting produced? So we will talk about all of that. So see what is happening. So most cellular reactive oxygen species are generated endogenously as byproducts of mitochondrial oxidative phosphorylation, or formed as intermediates of oxidoreductase enzymes and metal catalase. Okay, so where is catalase acting? Catalase is acting on hydrogen peroxide and converting it into water and molecular oxygen. Now this is further getting converted into different superoxide radicals, okay, reactive oxygen species. So hello, this is the last class, and in this today's class we are going to talk about a lot of different reactive oxygen species and other topics such as intrinsically disordered proteins. We are going to talk about protein sequencing and viruses. Okay, so there are miscellaneous topics for you and a lot of MCQ questions. Hopefully you're going to enjoy this class. Okay, now, so most cellular reactive oxygen species are generated endogenously as byproducts of mitochondrial oxidative phosphorylation, or as formed as intermediates of the oxidoreductase enzymes and metal catalase oxidation. So since oxygen atoms contain two unpaired electrons in separate orbits of its outer electron shell, it is susceptible to radical formation. The sequential reduction of oxygen through the addition of electrons lead to the formation of a number of reactive oxygen species, including superoxide anion, hydrogen peroxide, hydroxyl radical, hypochlorous acid, etc., and nitric oxide. Okay, so all of these different radicals are formed. So superoxide dismutase, or this particular enzyme that we are talking about. So superoxide dismutase catalyzes the dismutation of superoxide anion into oxygen or H2O2 and catalase catalyzes the decomposition of H2O2 into H2O and O2. Okay, so these two enzymes are very important. Now we will see what happens when ROS is produced. So our cell death can happen in a different form of ways. Okay, now if you look at all of these different ways in which the cell death is happening, you will see that in majority of these ways we will find the involvement of ROS. Okay, so under physiological conditions, ROS levels are carefully regulated where they operate as messenger in normal cell signal transduction, cell cycling, gene regulation and homeostasis. So oxidative damage of these biomolecules can trigger cellular apoptosis and is associated with aging and has been implicated in the pathogenesis in variety of diseases, cancers, neurodegenerative disorders as well as diabetes and inflammation. Okay, now what are the different things that happen during cell death and which of these include ROS? Okay, so autophagy. In autophagy, what is happening? ROS acts as a signal inducer and irreversibly oxidizes DNA and other critical cellular biomarkers, and then the cell is eaten up by its own enzymes, right? Now apoptosis. So we are all familiar with these terms. So what happens in apoptosis? So induced in inflammatory cells, that is neutrophil, macrophages, etc., eosinophils, leading to damaged DNA and protein. Next, where do we find the involvement of ROS? We see that there is paraptosis, there is pyronecrosis, there is pyroptosis. Okay, so all of these different pathways involve the activity of ROS and are assisting cell death. So that is why targeting ROS and helping and preventing this oxidative damage is one of the major targets of our therapeutic industry at this point, to prevent diseases such as cancer and neurodegenerative disorders, diabetes, inflammation, etc. Now, so we will see what do ROS do at high concentrations. Okay, so ROS are produced naturally as a result of normal cellular metabolism that we have established until now. Okay, so however, they can also be included in reactions to external stresses such as pollution, UV radiation, cigarette smoke, etc. Furthermore, some medicines and medical treatments such as chemotherapy can also cause oxidative stress to rise. Okay, and ROS at higher concentration results in oxidative damage which impacts lipids by causing chain breakage, increase in the membrane fluidity and permeability. Okay. In proteins, it is causing site-specific amino acid modification. Now you know how impactful all of these mutations are, right? So site-specific amino acid modification, fragmentation of the protein chain, aggregation of cross-linked reaction products, altered electric charge, enzyme inactivation, increased susceptibility of proteins to proteolysis. So all of these different conditions are pushing the cell towards death. There can be cancer, there can be other different diseases, different kinds of diseases in the body, and there also the oxidative stress is very high, and all of these different changes can happen in the cells, okay, because of this oxidative damage. So in DNA, you can see that there is oxidation of the DNA bases and the DNA sugar, strand breakage, removal of nucleotides, modification of bases, and protein DNA crosslinks can also get formed. Right. So next what we will talk about is cysteine mediated species. Okay. So cysteine contains a thiol group. We know that there is this SH group in cysteine which is highly reactive towards ROS. This makes cysteine one of the most commonly oxidized residues in proteins. So that is something very interesting. Okay. Now types of cysteine modification by ROS. So see what is happening. There is disulfide by bond formation. Okay. So ROS can oxidize the thiol group of the two cysteine residues leading to formation of disulfide bond, which can alter the protein structure and function. Then there can be sulfonic acid. One second. I am facing some connectivity issues. Okay. So sulfonic acid formation. So the thiol group can be oxidized to form sulfonic acid. So all the different structures are given in this figure for your understanding. Okay, sulfonic acid, which is reversible but highly reactive. Then sulfonic acid and sulfonic acid. So further oxidation results in the formation of sulfonic and sulfonic acids. These modifications are irreversible and result in permanent loss of function of the protein. Okay. Now this results in enzyme inactivation, disruption of enzyme structure, and regulatory inhibition of enzyme function. Now we have a question for you. Okay, no, we have a little more. So now we talked about cysteines and now we are going to talk about tyrosine. So tyrosines are also very important when it comes to ROS mediated protein damage. Okay, so tyrosines contain a phenolic hydroxyl group, okay, that is susceptible to oxidation by ROS, leading to the formation of various oxidative products. So type of tyrosine modifications by ROS: dityrosine formation. Two tyrosine residues can be cross-linked via covalent bond forming dityrosine. This can cause protein aggregation and loss of function. Okay. Then there can be 3-nitrotyrosine, that is peroxynitrite, a reactive oxygen species derived from ROS. This modification often affects the signaling proteins and can impair their function. Next is tyrosyl radical. So what is this? So ROS can induce the formation of a tyrosyl radical, which can propagate oxidative damage to the parts of the proteins and neighboring cell. Okay. So now the reactions are given for your understanding. So tyrosine can get converted into dityrosine by ROS. Okay. And then there can be other products that can be formed. So now we come to a question. So you can try to answer this question. Okay, so which one of the following oxidized form of cysteine is more likely to be directly linked to protein damage? Whether it is sulfonic acid, sulfonic acid or sulfanic acid. Okay. So we talked about this just now. You can answer in the chat box. D. Okay. D is sulfanic acid. Let's see. Achha. So the right answer is C, which is sulfonic acid form. So with the increasing levels of reactive species, cysteine is oxidized into sulfanic acid. It can further be oxidized to sulfonic and sulfonic acid, and this particular form, which is RSO3H, this is sulfonic acid, and this is considered to be an irreversible modification. It is directly linked with protein damage. Okay. Now, there is another question on tyrosine mediated damage that is caused by ROS. Okay. So dityrosine and nitrotyrosine are products from tyrosine in the presence of various reactive oxygen species. These two biomarkers are generally detected by dash and dash spectroscopy. So why is that so? We will talk about this in the upcoming slide. Okay, so exactly why these two different products are detected by two different kinds of spectroscopy. Okay, so you can try to answer this question in the chat box. Okay, we'll look at the answer, right answer. Okay, so the right answer is C. And if you see the wavelength and absorbance, you will see that tyrosine absorbs at what? Around 270 nanometer. Okay. Then nitrotyrosine, this is absorbing at around 350 nanometer. And then again nitrotyrosine at pH 10, this is again absorbing at around 430 or so nanometer. So now we will look at this in details. So in fluorescence spectroscopy, the intensity of dityrosine is enhanced with respect to tyrosine. Okay, so it can be detected by fluorescence spectroscopy. In UV visible spectroscopy, nitrotyrosine shows two absorbance peaks. So one is at 278 nanometer and the other one is 350 nanometer. So this is what we can see. Okay. So in dityrosine, what happens is the two peaks are at 278 nanometer and 350 nanometer. So that is how we can distinguish between these two. Okay, nitrotyrosine and tyrosine. Now we will look at this in a little more details. So nitrotyrosine forms when tyrosine undergoes nitration of the phenolic ring, typically through interaction with reactive nitrogen species like peroxynitrite. At 278 nanometer, nitrotyrosine shows an absorbance peak corresponding to its aromatic ring's electronic transfers, which is similar to tyrosine but slightly shifted due to nitration. At 350, nitrotyrosine exhibits a unique absorbance peak caused by nitro group, that is NO2 group, and attached to the aromatic ring, which is absent in the tyrosine. So this distinct peak allows for clear detection and quantification of the nitrotyrosine using UV visible spectroscopy. Okay, so tyrosine itself has some intrinsic fluorescence, but dityrosine forms due to the cross-linking of two tyrosines via ROS. Its fluorescence intensity increases significantly. So fluorescence intensity enhancement, the presence of cross-linked structure in dityrosine changes its electronic environment, leading to greater absorption of excitation light and stronger emission. Okay, this makes dityrosine easily detectable compared to tyrosine. Now, as a result, the fluorescence spectroscopy is highly sensitive to detecting dityrosine and can distinguish it from the normal tyrosine due to enhanced fluorescence. Right. So now that we have talked about how reactive oxygen species affects proteins and different residues in proteins, so now we are going to talk about protein sequencing. Okay, so that we are going to talk about two different methods. One is Sanger method of protein sequencing and the other one is Edman methods of protein sequencing. Okay, so what happens now? Both of these affect the N-terminal residue of the protein, and that is how the entire protein is sequenced. So amino terminal end of the polypeptide chain can be determined by three methods. One is Sanger method, Edman's method, and dansyl chloride method. Okay, so what happens is, in Sanger method, we have this fluorodinitrobenzene. Now, this compound comes and it attaches with the N-terminal. Then what happens? Acid hydrolysis happens and the free amino acid is released and the amino acid or the dinitrophenyl amino acid complex is released. So that is how you get the first residue at the N-terminal end. Okay. Then what happens is, in the dansyl chloride mediated process, once again, there is the dansyl chloride derivative of the peptide that comes. Okay. Now, this again undergoes hydrolysis and you get the N-terminal residue. So, in Sanger sequencing, fluorodinitrobenzene is used. Polypeptides react with this compound to form dinitrophenol complex with polypeptide. So, subsequent analysis releases a colored dinitrophenol labeled amino terminal amino acid, which is identified by its characteristic migration rate on thin layer chromatography or paper electrophoresis. Okay. Next, what happens in Edman degradation? So, in Edman degradation, what happens is phenyl isothiocyanate or PIT is the compound that is used. First, the polypeptide is reacted with phenyl isothiocyanate to form a polypeptidyl phenyl thiocarbamyl derivative, okay, which is this PTC derivative. And then what happens is gentle hydrolysis releases the N-terminal amino acids as PTH. So, phenyl thiohydantoin. So, which can be separated and detected spectrophotometrically. Up to this stage, the method is used to determine N-terminal amino acid, This method can also be extended to determine the polypeptide sequence. Okay, so for this, the release of the PTH and the remaining intact polypeptide shortened by one amino acid is then ready for further cycles of this procedure. Okay, so that is how this is done to get the sequence of the protein. Next is dansyl chloride mediated process. So here this method is more sensitive because of fluorescence measurement. So dansyl chloride is taken, it is labeled with the protein, and then acid hydrolysis happens, and then the dansyl alanine is formed and the amino acids are separated out. So now the question. So what is the question? So which of the reagents is not used for analyzing the N-terminal of a peptide sequence? Phenyl isothiocyanate, cyanogen bromide, 1-fluoro-2,4-dinitrobenzene, and dansyl chloride. So these are the four options that you have, and you have to tell me which one of these is not used for N-terminal peptide sequencing. Okay, so you can answer in the chat box. Okay, you can try answering. We just discussed this. Should not be difficult. We talked about 3 methods. We did not talk about one of the methods. So which one was it? Okay, we look at the answer. So the answer is, right answer is B, which is cyanogen bromide. Now N-terminal of the peptide can be analyzed by Edman degradation using PITC. Sanger's reagent uses this fluorodinitrobenzene or dansyl derivatives can be used for N-terminal detection. But cyanogen bromide cuts the peptide bonds at the C-terminal site of the methionine residue. So we have studied about this in our one of our previous classes, right? That cyanogen bromide is useful for cleaving, chopping off peptides from the C-terminal end. Now, we'll talk a little about viruses, okay? So, what is a virus? Of course, you know, now that COVID has, like we have encountered COVID already, everybody knows what's a virus now, right? So, virus is a non-cellular infectious entity made up of genetic material and protein that can invade into a host cell and reproduce only within the living cell of a bacteria, plant or animal using the host cell machinery. Okay. So now then, is the virus living or non-living? That is the question. So scientists have come up with the answer, and that is viruses are dead or non-living outside the host cell, and they are living inside the host cell. Okay. So there is no sign of life outside the host cell. It usually lacks a cell wall but is surrounded by protective protein coat called the capsid. It carries its genetic element, which is either RNA or DNA. Okay. Now, how does this look like? So, see, a virus that infects a bacteria is called a bacteriophage, and it is made up of capsid and the nucleic acid. Okay, the material, genetic material. Now, if we talk about HIV virus, okay, so human immunodeficiency virus, which is HIV. Now, this has a lot of different parts. What are these? So, it has a capsid, it has the genetic material, it has the envelope, it has envelope proteins and enzymes plus RNA. RNA is the genetic material here. Okay. So, now we will talk about all of this in details. So, see what is there in the life cycle of a virus. So, life cycle of a virus is very interesting because, see, this is your host cell. Host cell has its genetic machinery. Okay. Now, what the virus does is, the virus comes and it injects its genetic material into the host cell. Okay. Now, what happens is, so viral attachment and penetration. Now, so here we are talking about a phage. So, phage is a bacteriophage. Okay. So, that means it's a virus which is affecting a bacteria. So, it is releasing its genetic material inside the bacteria. Now what is happening? This genetic material can survive in two ways. One is it can either integrate with the genetic material of the bacterial host. That is one way of doing it. The other way of doing it is it can be present separately, okay, in the form of a prophage, okay, and it can be there. So what happens is, see, so this phage DNA becomes incorporated into the host genome, and this is called lysogeny. So it is existing, it is amplifying with the genome of the organism, and it is just there, persistent, okay. For years it can be like that. So what is happening? So this is called lysogeny, and the bacteria in which this viral DNA is integrated, this is called the lysogen. Now there is something called excision. So what is excision? This viral DNA can separate out from the bacterial DNA. Why is that happening? When the bacteria is not getting sufficient amount of nutrient, okay, then the virus will feel that I am going to die, because the bacteria might die. So then what it does is it separates itself from this bacterial nucleic acid. Okay, so under stressful condition, this gets separated. And then it goes into the lytic cycle. So what is lytic cycle? It immediately captures the entire machinery of the bacteria, starts producing its own copies, and then make sure that the viral DNA is packaged into viral protein. Entire virus particle is formed. Virus particle goes, breaks open the cell, and it leaves. So that is lysis. That is why the cycle is called lytic cycle. Lysis means breaking open of the bacteria. Okay, breaking open, that is lysis. Now see, excision happens from the bacterial chromosome along with a short piece of bacterial DNA. The DNA is then packaged into newly formed capsids. Capsids are the viral protein coat. Okay, so infection phage. Infection. So the phage containing both viral and bacterial DNA infects a new host cell. Recombination happens. Okay, these are also there, events that can happen. So this original bacteria's DNA also integrates with the new bacteria's DNA. There can be genetic recombination, variation. Okay, and this is a transgenic so transduction basically happened here. Now what happens? This is incorporated in the new cell. Now under stressful condition, this prophage is excised, and it can either be present in the form of a separate entity inside the bacterial cell, or it can be going into the lytic cycle. So the phage DNA replicates, new protein capsids are made, and then they are packaged into the phage capsid, and the newly formed bacteriophage particles, they are released, okay, from the bacteria. Now, this is the simple way of talking about it. Now, viruses are not simple. They are very, very clever, and they have been surviving for millions of years without a body, without any machinery, without anything, but then they are surviving. So that means you can understand that they are evolutionarily very advanced. So they know how to make sure that with the minimal things that they have, they should be able to keep on propagating, right? So how do they do that? So animal viruses do not always express their genes using the normal flow of genetic material, that is from DNA to RNA to protein. Some viruses have double-stranded DNA genome like cellular organism and can follow normal flow. However, others may have single-stranded DNA, double-stranded RNA, or single-stranded RNA genomes. So the nature of the genome determines how the genome is replicated and expressed as viral proteins. So if the genome is single-stranded DNA, host enzyme will be used to make the second strand. Okay, complementary DNA will be made, and thus producing the double-stranded DNA. So this double-stranded DNA can now be replicated, transcribed, and translated in a similar way as host DNA. Now, if the viral genome is RNA, a different mechanism is used. So So there are three types of RNA. One is double-stranded RNA, then there can be single-stranded RNA in positive sense, and there can be negative sense single-stranded RNA. So what is this positive sense and what is this negative sense? Okay, so we'll talk about that. Now, if the virus has single-stranded positive sense genome, it can be translated directly by viral proteins. Okay. Now, the viral genome single-stranded RNA acts like a cellular mRNA. So that is what positive sense is. Okay, so it is 5' to 3'. It is like a cellular mRNA and it can be directly translated into the protein. But if the virus, however, if the virus contains a negative sense genome, then the host ribosome cannot translate it, right? Because that is 3' to 5' compared to our normal mRNA. So then what is going to happen? Then the virus will have a special enzyme which is RNA dependent RNA polymerase. So this is going to make a complementary strand. So then it is replicated into the single-stranded positive sense RNA, and this enzyme is brought in by the virus and it can be used to make the single-stranded positive sense RNA from the original negative sense single-stranded RNA genome. Now, this particular enzyme is also an enzyme for replication of double-stranded RNA viruses because it uses negative strand of the double-stranded genome as template to create the positive strand of single-stranded RNA, right? So, this newly synthesized single-stranded positive sense RNA copies can be translated by cellular ribosome. So, there can be two enzymes. One is RNA-dependent RNA polymerase and also RNA-dependent what? That is all. RNA-dependent RNA polymerase is sufficient for interconversion between single-stranded RNA and your positive sense single-stranded RNA and negative sense single-stranded RNA. So now, in case of HIV, so what happens in case of HIV is retroviruses which are positive sense RNA virus like HIV carry a special enzyme called reverse transcriptase. So what is reverse transcriptase? This is RNA dependent DNA polymerase. Okay. So with a capsid that synthesizes a complementary single-stranded DNA copy using the positive sense single-stranded RNA genome as the template. So the single-stranded DNA is then converted into double-stranded DNA by host enzyme, which can integrate into the host chromosome and become a permanent part of the host. The integrated viral genome is called a provirus. So when it is integrating, it is called a provirus. The virus can now remain in the host for a long time. So HIV infection, once it happens, once the virus enters into the body, it can take up to 10 years before it starts expressing. It can take 20 years and it can stay in this latent stage. Okay. Then the provirus stage is similar to the prophage stage in the bacterial infection during the lysogenic cycle. However, unlike prophage, provirus does not undergo excision after splicing into the genome. So see what is happening. This viral DNA is transported. Okay, so what is happening? The HIV fuses into the host membrane, and what happens? The HIV RNA reverse transcriptase enzyme integrase and other viral proteins enter into the cell. So the viral DNA is formed by reverse transcription in the cytoplasm. Viral DNA is transported across the nucleus and it enters into the host DNA. It joins with the host DNA as provirus. So new viral RNA is used as a genomic RNA to make viral proteins. Okay. Then new viral RNA and proteins move to the cell surface and newly formed mature HIV particles are made. The virus matures when the protease releases and the proteins form the proteins are released from to form the mature HIV particle. Okay, and it is released for further infection. Now, Which of the following processes do not happen in the HIV? Replication, reverse transcription, translation, and transcription. So which of these are not happening in HIV? Okay. You can answer in the chat box. Which of the following processes are not going to happen in HIV? Okay, so we look at the right answer. It is C, 1 and 4. So replication, replication is not going to happen, and transcription is not going to happen in an HIV. So in HIV, reverse synthesis of the DNA from the RNA is performed by enzyme reverse transcriptase. Translation is the synthesis of protein from mRNA. Since mRNA carries instruction for making new viral proteins, each section of the mRNA relates to making a different part of the HIV virus. Okay. Oh, so one of the very interesting things about viruses is that it can be, so for us, for humans, one single mRNA makes one single protein. Okay. But there what happens is, there one single mRNA can have, it's a polyprotein mRNA, and so it can make many proteins from the same mRNA. Okay. Now we are going to talk about a very deadly virus, and that is Ebola virus. Okay. So E virus is a filamentous shaped virus with dimensions of 800 nanometers long and 80 nanometer diameter, and it has an encapsulated single-stranded negative RNA. So there are 7 expressed proteins by Ebola. What are these? Glycoprotein. Then there is membrane-associated protein VP40, VP24, nucleocapsid. So what is a nucleocapsid? So nucleic acid plus the capsid together makes up the nucleocapsid. Okay. And then there is VP30, which is a phosphoprotein involved in RNA transcription. There is the viral genome. Viral genome is single-stranded negative sense RNA. And VP35, which inhibits antiviral immune response. And the polymerase, which is the L protein. So enzyme that catalyzes RNA replication. Okay. Now, why is this so important? Because Ebola virus still does not have a vaccine to cure it. And a lot of people are dead every year. So Ebola is incurable and deadly. The outbreak in West Africa in 2014 was unprecedented, dwarfing other human Ebola epidemics in the level of mortality. So around 24,600 or so people died. Oh, sorry. So these people were affected, and around 10,000 or so people died. So no approved treatment or vaccines for Ebola are available until now. Okay. Now the roles for these proteins are summarized as below. So NP is essential for, so all the short forms are given there. So protein. So NP is nucleoprotein. It is essential for RNA encapsulation. GP is glycoprotein, which is important for attachment of the virus to the host cell membrane, entering the nucleocapsid of the virus to the host cytoplasm. Then VP24, VP30, all of these have different functions. You can go through all these different functions. Okay. We'll try to solve one question. So now you will have to read about different viruses. I just covered around 2 to 3 viruses here, but it's very important that you read about all the viruses that are commonly there. Okay. Now, this is a question. So, which of the following statements is true about Ebola virus? So, the nucleocapsid of the Ebola virus contains a DNA genome. The lethal virus is made up of 7 proteins encoded by its genome. Matrix proteins often help to adopt different shapes of viruses for driving the budding process. The nucleocapsid of the virus is circular in nature. So you can try to answer. Which of the following statements is true about Ebola? we'll look at the right answer. It is option number C, 2 and 3. So the nucleocapsid is cylindrical in Ebola, not circular, right? So 4 is not correct. Now the cylindrical nucleocapsid contains RNA genome. That means 1 is not correct. It is not a DNA genome, it is an RNA genome. The genome encodes 7 proteins that get assembled to produce the deadly virus. Matrix proteins give shape to the virus and drives the process of budding. Okay. Now we will talk about proteins and their cleavages. Okay. Now we have already been practicing these questions. We'll practice them a little bit more because these are very important for your exams. You saw in the last class that there were around 3 to 4 questions in the same paper, and they were all from this particular topic of protease digestion, right? So an unknown protease cleaves the peptide sequence this into 3 polypeptides. The name of the specific protease is, so the protease is not very unknown, you are supposed to identify. So see where the protease is cleaving and whether it is taking into consideration the presence of proline or not, right? So that is something that you'll have to remember and tell me what is the name of the protease. So it is definitely an endopeptidase, right? It's not an exopeptidase. Okay, so we look at the right answer. The right answer is chymotrypsin. So chymotrypsin selectively cleaves at the carboxyl site of the aromatic residues W by F. Hence, the peptide cleaves the amide bond between the phenylalanine and proline, and it also cleaves the tyrosine and glycine, between tyrosine and glycine. Okay. Now what you need to consider is whether proline is present on the C terminal or the N terminal of the cleaving site. Yes, that is something that you need to consider. Now the polypeptide. So what is it doing? So this is the polypeptide chain and it is getting cleaved by thrombin and trypsin, which are the digested fragments. So what will happen once thrombin is used for digestion and then trypsin is used for digestion? So what are the different fragments that are going to be formed? Okay. For the second question, a tryptic digest, which means digested by trypsin, of an octapeptide contains 2 lysines, 2 methionines, 1 serine, 1 proline, 1 glycine and 1 arginine. Okay. Yielded 2 tripeptides and 1 dipeptide. Both of the tripeptides do not contain arginine. Which one of the sequences is shown correctly? So that is very important. This is how you will have to, this is the kind of questions that you can expect in your exams. Okay, so the entire composition of the amino acids is given and they're asking you what will be the original sequence of the polypeptide, if these are the different fragments that can be formed, okay, by the digestion of the 2 enzymes. Okay, now we'll try to solve these. Okay, so detailed solution is given for you. So since thrombin cleaves the carboxylic side of the arginine and trypsin cleaves the carboxylic side of both lysine and arginine, residues not followed by proline, digested fragments are this and this respectively. And for the second one, trypsin cleaves the carboxylic side of both lysine and arginine residue not followed by proline. So after tryptic digestion, peptide fragments are this and this. So both tripeptides do not contain arginine. So the final result would be option number B, which is the right answer. Okay. Now we will talk a little about intrinsically disordered proteins. Okay. So intrinsically disordered proteins are very interesting because normal proteins, they are folding normally, but some proteins are very disordered in nature. So they are having these flexible regions. These flexible regions are very interesting because they contain this short linear motifs. Okay, so in this flexible region there are tiny sequences, tiny motifs which help in protein-protein interaction and interaction with other components as well, okay, other biomolecules. So what are these different disordered proteins and why are they so important, right? So disordered proteins are very important because they are helping in different kinds of protein-protein interaction and protein-DNA interaction. Okay, structured proteins are definitely important, but intrinsically disordered proteins can be targeted as therapeutics for different kinds of diseases. A lot of transcription factors are also intrinsically disordered proteins. Now, why is it so important to study them? Because you don't have the crystal structures of these intrinsically disordered proteins. Okay, because disordered regions are very difficult to crystallize. So that is why NMR structures are there, and now you can use AlphaFold and all these different models, these softwares for getting the structures of these intrinsically disordered proteins, but then a lot of times these structures are not accurate. Okay, and that is why when you're targeting these as a therapeutic target, it is very difficult. That is why studying and understanding intrinsically disordered proteins is very, very interesting. Now, the term fly casting comes from the analogy of a fisherman casting a fly fishing line into the water. So a loose extended, so what happens is in that you can imagine that the part that the fisherman is holding, it is a rigid structure, and this extended region, it can go as far as you throw it, right? So that is why these intrinsically disordered regions are so important, because they are thermodynamically feasible to extend as much as they want, to go into any conformation that they want and make the interactions with other proteins as they want. Okay, so a loosely extended line can cover a more area than a coiled or the stiff line, making it easier to catch fish. Similarly, intrinsically disordered regions or intrinsically disordered proteins with unfolded or partially disordered regions can explore a large area of conformational space while searching for their tight binding partner. So that is one of the very interesting things about them. Next is the increased capture radius, the one that I told you. So disordered state of an IDP allows it to extend and sample a larger area, increasing the likelihood of its encountering its target. This is analogous to the fishing line extending further and covering more space. Then there is rapid binding. Okay, so once the IDP comes into close proximity with its target, it can rapidly collapse and fold into a specific structure. So I told you that these IDP regions, they have small motifs where the interaction happens, and these motifs are very important because these motifs can quickly fold into a specific helix or any other structured region and help in binding with this DNA or protein. Okay. Now, intrinsically disordered region can go into the minor groove of DNA and make very strong contacts. That is how they are very useful in transcription factors. Okay. Now, there can be, this mechanism is called induced fit or folding upon binding. This leads to an efficient interaction. Okay. Now, an example of fly casting mechanism can be found in the interaction between transcription factors and DNA. Many transcription factors have intrinsically disordered regions that help them to locate specific DNA sequences. The extended flexible structure of the transcription factor allow it to scan large regions of DNA. Once the correct binding site is found and the disordered region folds into a stable conformation, forming a strong interaction with the DNA. Right? So now we have a question for you. So which one of the following amino acids is preferably present in the intrinsically disordered proteins and play a role in preventing the formation of a stable structure? So now which of these amino acids are useful in ensuring that the disordered region in the protein is disordered? Okay. Which do you think is very important? Oh, I think the answer is already there. So, so it's not a question anymore. We just discussed this. Okay, so the right answer is glutamic acid, proline, and arginine. Why is that so? Because proline, when that is present in a peptide, it will disrupt the secondary structure. Also, glycine is a very good important residue for making it disordered in the presence of a polar amino acid. Stable non-polar core structure gets prevented. Hence, the option A matches all these criteria. Okay. Next, we have two more questions. Which among the following is not true about intrinsically disordered proteins? Okay. So you can answer in the chat box. Option A is intrinsically disordered proteins lack stable three-dimensional structure. IDPs are disordered only in localized regions. IDPs carry low density of charged amino acid residues. IDPs exist as highly flexible conformations. So which one is incorrect for the fly casting mechanism of IDP? So specific binding sites searched for and found while folding. Reaches high affinity formation. Non-specific binding sites searched for and found while folding. Easy sampling of many conformations. Which one of these do you think is incorrect? Okay, so you can answer in the chat box. Okay, since you are not answering, we'll look at the right answer. So the right answer is for question number A, the right answer is C. Okay, so IDPs carry high density of polar and charged amino acid residues such as lysine, glutamine, serine, arginine, and glutamic acid. These are the different residues that are largely present in IDPs. Okay. Now, which one is incorrect for the fly casting mechanism of IDP? So, non-specific binding is wrong. So, for fly casting, IDP specific binding site directed search and finding happens, and then it folds into its respective structure. Okay. Now we have another question. So now we have talked about PCR previously, right? And you know the different stages of PCR. So now this is an interesting question in which they are telling you that during the molecular cloning experiment, the ligation step. What is ligation step? This is the vector and you are ligating the vector with the insert. You have cleaved the two ends of the, you have digested the two ends of the vector with two different restriction enzymes, and you have digested the two ends of the insert with two different restriction enzymes, and you want them to ligate, right? They are having these sticky ends. So now this ligation reaction is normally performed at 16 or 22 degrees centigrade rather than 37 degrees centigrade. But for a T4 DNA ligase, the optimal temperature is actually 37, right, for enzyme activity. Then why is it that, what is the primary reason for using lower temperature during ligation? So this is a very practical question and this is something that you need to know. So there are 4 points. What can the answer be? The first option is to prevent denaturation of the enzyme at higher temperature, to reduce ATP degradation in the reaction mix, to increase the stability and annealing of the complementary sticky ends during ligation, to enhance the kinetic energy of the DNA ends for efficient ligation. Okay, so which one do you think is the right answer? A. Okay, great. Okay, so the right answer would be C. So at lower temperatures, that is 16 to 32 degrees centigrade, cohesive ends means sticky ends, can stably anneal due to reduced thermal motion. So when you are increasing the heat of the system, of course the kinetic energy of the molecules also increase. So they are going to come close by and they are going to encounter one another quickly, but they're also going to get dissociated quickly, right? So what happens is you need to make sure that thermal motion is kind of reduced, allowing this is kind of a compromise between the optimal activity and the optimal temperature. Okay, so T4DNA ligase effectively forms phosphodiester bond between two adjacent nucleotides. So at 37 degree centigrade, DNA ends are more likely to dissociate before ligation occurs. So that is something that you need to remember. Okay. Now, both T4 DNA ligase and T4 polynucleotide kinase. So what is this kinase enzyme? It is adding the phosphate group at the 5' end, which is necessary for ligation process, right? So this PNK enzyme, polynucleotide kinase, requires specific reaction buffer for optimal activity. So what is the main function of these buffers in these respective reactions, right? So what is there in this buffer anyway? So A is to maintain the constant pH and provide cofactors necessary for enzyme activity and catalysis of phosphodiester or phosphate bond formation. B is to denature DNA temporarily, allowing the enzymes to access single-stranded region. C is to act as crowding agents. So what is crowding agents? Crowding agents ensure that two different molecules meet and they are just there to have more molecules in the system and make more collisions so that the two required molecules meet. So crowding agents that increases the local concentration of the DNA molecules to inhibit the exonuclease containing exonuclease contamination during the reaction. Okay, so why do you think these buffers are necessary for the enzyme activity? A, right, that is correct. A is the right answer. So see, why is it the right answer? That is because both ligase and kinase reactions require ATP and Mg2+ as cofactors. The buffer not only maintains an optimal pH environment for catalytic residues, but also supplies divalent cations that stabilize the enzyme substrate complex. Okay, so in ligation, this facilitates phosphodiester bond formation between DNA ends, while in kinase reactions, it enables transfer of the gamma phosphate from ATP to the 5' hydroxyl end of the DNA or RNA. Okay, now for your understanding, I have given the entire composition of the ligase buffer and kinase buffer and what is the role of each component. Okay, for example, tris maintains pH, MgCl2 provides Mg2+ for as the cofactor. Then you have dithiotritol. Why is that important? Because it helps to maintain the reduced state of the ligase enzyme. Okay, these are all small questions which can be there for your exams. So small concepts that you need to remember. ATP, of course, you know that this is an important energy source for the ligase reaction. Okay, so the entire product that is getting formed, which is getting transferred, is containing AMP. Now we have BSA. Why is BSA there? To stabilize the enzyme during the reaction. Okay, so that is why it is very important. Now this buffer supports optimal ligation conditions, enabling DNA fragments to be efficiently joined together. And the kinase buffer, this has tris and MgCl2 and it has DTT and ATP. Why is this so important? Because this helps in phosphorylation of the substrate, commonly used in reactions during the PNK and PNK step to add phosphate group to DNA or RNA ends. Okay, so after you perform a PCR, polymerase chain reaction, it is not having these phosphorylated ends, right? So that is why you are supposed to add them. Now, that is all for today. That is all for this semester. Okay, it was great spending so much time with all of you, and I got to learn so many things from you, and hopefully you got to learn something from the classes. Okay, thank you so much for your participation. So if you do not have any more questions, we'll end the chapter and the course today. Okay, all the very best for your exams. If you have any doubts regarding anything, you can reach out to me. You already have my mail ID, I guess, and on the NPTEL platform also you can reach out and you can ask me the questions and answers, right? So thank you so much to all of you. Okay. So I'll be ending the call now if you do not have any more doubts or anything. Okay. Have a very nice time ahead. All the best for your future endeavors. All right.